So let's differentiate between the prokaryotic and eukaryotic cells. So first of all, I will be starting with the outer covering of the cell. So outer covering of the cell, it is represented by the cell wall. In the case of the prokaryotic cell, there is presence of non-cellulosic cell wall. Non-cellulosic cell wall is present, but some of the exceptions, they are also there. One of the exception is mycoplasma. Around the mycoplasma, the cell wall is absent. Now coming to the eukaryotic cell, it is very well known to you. अगर plant होगा तो cellulosic cell wall present होगी. Protist होंगे, again the cell wall is present. Fungi, cell wall present, and in the case of the animals, the cell wall is absent. So after this, now coming to the next difference. So cell wall के नीचे जो layer present होती है, that is very well known to you. That layer is plasma membrane. Yes. So below the cell wall, the covering which is present, that is the plasma membrane. Prokaryotic cell ho चाहे eukaryotic cell ho, the plasma membrane, in both of them they are present, and it is very well known to all of you that the plasma membrane, it consists of lipid bilayer, and along with that there is presence of proteins. Lipid bilayer and protein, that constitutes the true membrane, and this membrane is present both in the prokaryote as well as the eukaryotes. Eukaryotes के अंदर जो cytoplasm present है, in the cytoplasm there is also presence of membrane bound organelles. There is presence of membrane bound organelles. The membrane bound organelles like endoplasmic reticulum, mitochondria, Golgi bodies, chloroplast, vacuole, lysosome. So all these membrane bound organelles, they are present in the eukaryotic cells. Now साइटोप्लाज्म के अंदर भी मेम्ब्रेन बाउंड ऑर्गेनेल्स प्रेजेंट है और साइटोप्लाज्म के बाहर भी मेम्ब्रेन प्रेजेंट है, दैट इज प्लाज्मा मेम्ब्रेन. तो इसका मतलब यूकैरियोटिक सेल के अंदर प्रेजेंट है डबल मेम्ब्रेन. There is presence of double membrane system, and this double membrane system, it can simply be known as two envelope system, right? But let's see what is the scenario in the case of the prokaryotes. Prokaryotes के अंदर plasma membrane तो present है, but the membrane bound cell organelles, they are absent. So it means in the prokaryotes now there is presence of only single membrane. Yes, there is presence of only single membrane. So now this can simply be known as single or one envelope system. Now coming to the next point. Membrane bound organelles. In the prokaryotes, they are absent, but please keep this in mind that the non-membranous organelle in the prokaryote is absent, and I have discussed in the previous chapter that this organelle is ribosome. Yes. So can you tell me what is the function of the ribosome? Ribosomes, they are involved in the synthesis of the protein. That's why they are known as protein factories. And the type of ribosomes which are present in the prokaryotes, that is 70S. 70S राइबोसोम प्रेजेंट है प्रोकैरियोट के साइटोप्लाज्म के अंदर. In the case of the यूकैरियोट्स के अंदर जो साइटोप्लाज्म में प्रेजेंट है 80S टाइप ऑफ राइबोसोम्स, and in the organelles like mitochondria and chloroplast, there is presence of 70S type of ribosome. तो साइटोप्लाज्म के अंदर प्रेजेंट है 80S, बट ऑर्गेनेल्स के अंदर जो राइबोसोम प्रेजेंट है दे आर दी 70S type. Now I had discussed in the previous classes in the chapter biological classification that in the case of the prokaryotes, this plasma membrane, it shows ingrowth. Yes, can you tell me what these ingrowths they are known as? These ingrowths they are known as mesosome. So let's write this information. So let's discuss now the next point. So now the next point, that is mesosomes. तो ये बैक्टीरियल सेल के अंदर मैंने ये बनाई है सेल वॉल, राइट. Inner to the cell wall there is presence of the plasma membrane. Plasma membrane, and you can see here the ingrowths of the plasma membrane. It's very well known to all of you that these ingrowths they are known as mesosomes. Now one of the function of the mesosome is that they are involved in the DNA replication, because in the case of the prokaryotes the DNA, the genetic material, it is attached with the mesosome. तो एक फंक्शन क्या हुआ, डीएनए रेप्लिकेशन के अंदर हेल्प करेगा, एंड द सेकंड वन, द मेसोसोम्स दे आल्सो पोजेस respiratory enzymes. Yes, some of the respiratory enzymes, they are associated with the mesosomes, but in the case of the eukaryotic cells, these ingrowths, they are absent. Now, these were the ingrowths of the plasma membrane. Right? Now, let's discuss about the outgrowth of the plasma membrane. We have discussed that the outgrowth of the plasma membrane, they are villi, and the another one is fimbriae. Villi and fimbriae. Now what they are going to represent? They are the outgrowth of the plasma membrane. Like this. Like this. So I am not making all the villi and fimbriae outside the cell. Now ये जो outgrowths हैं, you know they are made up of the protein. That protein is known as pillin protein. And one of the function common for these two structures, that these structures they help in the attachment. Yes, the structure they help in the adhesion. Pili and fimbriae. In the prokaryotic cells they are present, and in the case of the eukaryotes these are absent. Now next feature. The next feature which I have to discuss here, that is vacuole. Just now I had discussed that in the eukaryotic cell the membrane bound organelles they are present, and one of the membrane bound organelle present inside the cell, that is the vacuole. तो ये मैंने बनाया eukaryotic cell, और इसके अंदर अब मैं ये वैक्यूल दिखा रही हूं. दिस इज अ वैक्यूल, और ये है आसपास साइटोप्लाज्म. Now in the eukaryotic cell, the cytoplasm, it shows movement. Yes, इस वैक्यूल के आसपास साइटोप्लाज्म मूवमेंट शो करेगा, क्लॉकवाइज डायरेक्शन के अंदर या एंटी क्लॉकवाइज डायरेक्शन के अंदर. द मूवमेंट ऑफ साइटोप्लाज्म अराउंड द वैक्यूल, दिस इज नोन एज साइक्लोसिस, और इट इज नोन एज साइटोप्लाज्मिक स्ट्रीमिंग. Right? So movement of the cytoplasm around the vacuole, this is cyclosis. Now what is the condition with respect prokaryotic cell? Prokaryotic cell के अंदर the true vacuole is absent. Membrane bound organelle is absent है, तो true vacuole absent है. There is presence of gas vacuole. There is presence of vacuole which is filled with gases, and the function of this gas vacuole, it helps in the floating of the prokaryotic cells. Yes, it will provide the buoyancy, but this gas vacuole is not known as true vacuole because it is not surrounded with the help of true membrane. This is not surrounded with the help of true membrane. So what is the definition for the true membrane? True membrane is one which is made up of the lipid bilayer and proteins, but here the membrane which is present, that is only proteinaceous in nature. So you're getting this point? ट्रू मेम्ब्रेन किसको कहेंगे? जिसके अंदर प्रेजेंट होगा लिपिड बाइलेयर एंड प्रोटीन. बट हियर द मेम्ब्रेन विच इज प्रेजेंट अराउंड द वैक्यूल, दैट इज only proteinaceous membrane. That's why these vacuoles, they are not true vacuoles, or simply you can call them as pseudo vacuoles, right? So gas vacuoles, they are pseudo vacuoles, and in the case of the prokaryotic cell, the streaming, cytoplasmic streaming is absent. Now the next difference. So let's see the more differences between the prokaryotic and the eukaryotic cells. Now here I will be discussing genetic material, the organization of genetic material. Now in the prokaryotic cell there is presence of primitive nucleus. Primitive nucleus present है जिसके अंदर the genetic material is not surrounded with the help of the membrane. So here genetic material is naked, or you can say that genetic material, it is not surrounded by membrane. तो प्रिमिटिव न्यूक्लियस जो प्रोकैरियोट्स के अंदर प्रेजेंट है, दिस प्रिमिटिव न्यूक्लियस, इट इज नोन एज न्यूक्लियोइड. इट इज ऑल्सो नोन एज जीनोफोर. इट इज नोन एज प्रोक्रोमोसोम. or it is also known as incipient nucleus. तो जो प्रिमिटिव न्यूक्लियस प्रोकैरियोट्स के अंदर प्रेजेंट है उसके ये सारे ऑल्टरनेटिव नेम्स हैं. So let's see what is the scenario with respect to the eukaryotes. Now in the case of the eukaryotes, now the genetic material is surrounded with the help of the membrane, nuclear membrane. तो eukaryotes के अंदर प्रेजेंट है well developed nucleus जहां पर जेनेटिक मटेरियल is enclosed by nuclear membrane. In the case of the eukaryotes, nucleus के अंदर present है एक structure that is nucleolus. Yes, here the nucleolus is present. And in the case of the prokaryotes, nucleolus is absent. Now let's see what is the condition of the genetic material in the prokaryotes. जो DNA prokaryotes के अंदर present है, this DNA is double stranded, that is DS, double stranded DNA, and this DNA is circular. Circular का मतलब the two ends of the DNA they are joined. DNA is closed. This DNA is coiled. This DNA is coiled और coiling के अंदर जो प्रोटीन हेल्प कर रहे हैं, these proteins they are polyamines, right? In the eukaryotes again there is presence of dsDNA. But now you please keep this in mind. Now this DNA is linear. Linear DNA. Now this DNA again it is coiled, and for the coiling the proteins which are involved they are the histone proteins. तो ये बात याद रखेंगे, यूकैरियोटिक सेल्स के अंदर कॉइलिंग में हेल्प करेंगे histone proteins, but in the case of the prokaryotes there is involvement of non-histone proteins, and these proteins they are known as polyamines. In the case of the prokaryotes, the DNA is present only in the cytoplasm. DNA सिर्फ साइटोप्लाज्म पे प्रेजेंट है, बट यूकैरियोट्स के अंदर द DNA is present in nucleus. Along with that, the DNA is present in mitochondria as well as chloroplast, right? Next feature, let's see what is the position, what is the area of transcription and translation. अब जो प्रोकैरियोटिक सेल है, इस प्रोकैरियोटिक सेल के अंदर सिर्फ एक ही कंपार्टमेंट है, दैट इज साइटोप्लाज्म. The membrane bound organelles in the prokaryotic cell they are absent, so everything is going to occur in that single compartment. So transcription and translation they will occur in the single compartment, that is the cytoplasm. Transcription is very well known to you. What is transcription? That is DNA के अंदर present है coded information. This coded information is utilized for the formation of the RNA. Formation of the RNA from the DNA, this is known as transcription. And this RNA has information for the formation of proteins. Formation of the proteins by utilizing the coded information on the RNA. This is known as translation. So this is transcription, this is translation, and in the case of the prokaryote, this process, these processes, they will occur in the cytoplasm. And what is the condition in the eukaryotes? यूकैरियोट्स के अंदर जो ट्रांसक्रिप्शन है, the transcription, it will occur in nucleus, right? RNA बनेगा न्यूक्लियस के अंदर, RNA न्यूक्लियस से ट्रेवल करेगा और पहुंचेगा साइटोप्लाज्म के अंदर, एंड इन दी साइटोप्लाज्म translation will occur. Getting this or not? तो ट्रांसक्रिप्शन कहाँ हुआ? आरएनए कहाँ पर बना? न्यूक्लियस में. ये जो आरएनए ये न्यूक्लियस से ट्रेवल करेगा, पहुंचेगा साइटोप्लाज्म में, एंड इन द साइटोप्लाज्म विद द हेल्प ऑफ द राइबोसोम translation will occur, which results in the formation of the protein. So it means in the case of the eukaryotes, so the transcription occurs in nucleus and the translation occurs in cytoplasm. पर इसके साथ साथ अभी मैंने आपको बताया है कि डीएनए सिर्फ न्यूक्लियस में नहीं है, it is also present in the mitochondria as well as chloroplast. So it means mitochondria and chloroplast, they will also show transcription as well as translation. So this point is very much clear to all of you. Now after this, now the next point which I will discuss here, that is flagella. So let's discuss the another feature. That is flagella. It is very well known to all of you that the flagella, this is a locomotory structure which helps in the movement of organism, movement of cell. Prokaryotic cells के अंदर जो flagella present है, that flagella is made up of flagellin protein, and in the case of the eukaryotes, flagella is made up of tubulin protein. And in this chapter, I will be discussing the detailed structure of the eukaryotic flagella. You please keep this in mind that the eukaryotic flagella, it shows 9+2 arrangement. Now the next one, that is cytoskeleton. अब जो यूकैरियोटिक सेल है, इसके अंदर प्रेजेंट है एक फ्रेमवर्क. यस, वन स्केलेटन, वन फ्रेमवर्क इज प्रेजेंट. दिस फ्रेमवर्क इज फॉर्म्ड ऑफ प्रोटीनेशियस ट्यूब्यूल्स एंड फाइबर्स. तो यूकैरियोटिक सेल के अंदर प्रेजेंट है स्ट्रक्चरल फ्रेमवर्क. दिस स्ट्रक्चरल फ्रेमवर्क इज ऑफ proteinaceous tubules and fibers. तो यूकैरियोटिक सेल के अंदर साइटोप्लाज्म में ये फ्रेमवर्क प्रेजेंट है. ये जो फ्रेमवर्क है ये काम करेगा स्केलेटन का. इट विल प्रोवाइड दी सपोर्ट. So this framework which will provide the support to the cytoplasm, this is known as cytoskeleton. Yes, so this is cytoskeleton. Eukaryotic cell के अंदर ये skeleton present है, and in the prokaryotic cell this network, this framework is absent. Next feature, that is spindle formation during the cell division. In the case of the prokaryotes, in the case of bacteria, one of the method of the asexual reproduction is binary fission. तो इट्स वेरी वेल नोन टू यू, बाइनरी फिजन के अंदर एक सेल डिवाइड करेगा और बनाएगा दो डॉटर सेल, बट ड्यूरिंग द डिवीजन इन द प्रोकैरियोटिक सेल स्पिंडल फॉर्मेशन इज एब्सेंट. स्पिंडल फॉर्मेशन एब्सेंट है, बट यूकैरियोटिक सेल के अंदर डिवीजन के टाइम पर स्पिंडल फॉर्मेशन प्रेजेंट होगी. प्रोकैरियोट्स के अंदर जब सेल डिवीजन होती है तो स्पिंडल फॉर्मेशन एब्सेंट है. दैट्स व्हाई दिस डिवीजन व्हिच इज अकरिंग, इट इज नोन एज ए माइटोटिक डिवीजन. नाउ कमिंग टू द सेक्सुअल रिप्रोडक्शन. In the eukaryotic cells, the sexual reproduction is present. But when we talk about the bacteria, prokaryotic organism, the true form of sexual reproduction is absent. तो ये मैंने पहले चैप्टर के अंदर डिस्कस करा है इन द बायोलॉजिकल क्लासिफिकेशन कि बैक्टीरिया के अंदर गैमीट फॉर्मेशन, गैमेटिक फ्यूजन, मियोसिस, ऑल दीज थिंग्स, द स्ट्रक्चर फॉर्मेशन इन द प्रोकैरियोट्स इज एब्सेंट. पर प्रोकैरियोट्स के अंदर सिर्फ होगा रिकॉम्बिनेशन. Recombination will occur, that is there will be transfer of genes from one bacteria to other bacteria. So this is not a true form of sexual reproduction. Instead, it is known as parasexual reproduction. So the important points associated with the prokaryotic and eukaryotic cell is very much clear to all of you. तो यहाँ पर जो हमने कुछ important point discuss करा है, one, that in the case of the prokaryotes there is presence of the single envelope system, and in the case of the eukaryotes double envelope system. If we talk about the genetic material, in the prokaryotes it is naked without the membrane, and in the eukaryotes the membrane, nuclear membrane is present. The coiling of the DNA with the help of the histone protein, this is the feature of the eukaryotes, and in the prokaryotes the coiling is assisted with the help of the polyamines. So after this, now I will be starting with the detailed structure of the eukaryotic cell, and in the eukaryotic cell I will be dealing with the outermost covering which is present in the plant cell, that is the cell wall.